So our next speaker is Dr. Kelsey McIntyre, who is on faculty here at the University of Minnesota. She's double boarded in cytogenetics and clinical molecular genetics and genomics, and her expertise in both of these areas have been very helpful as she's co-led our efforts to develop a whole genome sequencing pipeline calling structure variation and copy number variants in particular. So please.
Yes, so today, thank you for the introduction. So I'm going to talk about a validation project that we've been working on with the Molecular Diagnostics Lab to validate whole genome sequencing for detection of structural variants. I have no disclosures. So this is our genome and what it contains, and interestingly, there is a lot of information within the genome that isn't what we think of as being very exciting clinically. So a lot of the genome is made up of retrotransposable elements, repeat sequences, and introns, and only about 1 to 2% of the genome comprises the exome, and the exome is all the exons from the approximately 20,000 genes. However, the exome contains approximately 85% of all the known disease-causing variants. So when we talk about structural variants, there are a number of classes here, but one thing to just point out is when I say structural variants, I mean things that are greater than 50 base pairs, and this distinguishes from indels that would be small deletions and duplications. So we're really talking about larger things here. That can include deletions, novel insertions, mobile element insertions, duplications of different types, inversions, and translocations. I also may kind of inversely use the word copy number variants in the same breath as I'm using the term structural variants, although formally copy number variants are larger, being at least 1 kb in size, and include only things that change the copy number state. So gains or losses, deletions or duplications, does not include balanced events. But we're really including all of these types of events here in what we are looking for. So in terms of clinical detection of structural variants, there are lots of clinical assays out there that already detect structural variants, but there is sort of a gap here where we think that we can provide an added benefit. So when you think about the large events, you think about conventional cytogenetics, so karyotyping, FISH, and array CGH. Now, karyotyping is going to be limited, of course, to very large events that are visible under the microscope when visualizing metaphase spreads. Here, this graph is showing it down to 1 megabase, although I would argue that you would be a pretty amazing cytogeneticist if you could actually see something that was 1 megabase. In reality, it's probably more like 5 to 10 megabase resolution of karyotyping. FISH will get you a little bit better, but it's a targeted assay, so you have to know exactly what you're looking for and is not practical on a genome scale. Array CGH is genome-wide. However, it does really have a limitation based on probe spacing, and it will get you down to about 25 kb. There are more specific arrays that look at exon level or smaller deletions and duplications, getting down to maybe 10 kb, but those are typically for targeted assays looking at specific disease types and specific gene sets. MLPA will cover the entire bandwidth of small events up to very large chromosomal level events. However, that also is a targeted assay and is really not feasible at all on a genome scale. And then we have our sequencing assays. So that can include Sanger sequencing, targeted panels, NGS, whole exome, without any specific bioinformatic algorithms or tools to interrogate structural variants. Your general sequencing will get you up to about 100 base pairs. And after that, you're gonna potentially have some deletions, duplications, insertions, and things that are just gonna drop out and be missed by your typical calling. qPCR, similar to FISH or MLPA, is a targeted assay. So we really have this gap right here in the middle, which, interestingly enough, overlaps with the gene and exon size, where we have a lot of clinical interest in events that are between 100 base pairs or even up to 1 kb to 10 kb, but we don't have a good clinical assay that we can interrogate the entire genome for structural variants of this size. So just to talk a little bit about the differences between exome and genome sequencing, that's important for why we are going for genome sequencing with this project and not with exome, is related to kind of some of the technical differences. So with exome sequencing, you have your genomic DNA, you fragment that DNA and ligate your adapters. And then at this point, you have to capture your exome. So you don't want to waste a lot of your sequencing reads on parts of the genome that you're not actually trying to sequence. So you add capture probes and you pull out just the exons. Then you have to amplify and sequence in a line. With whole genome sequencing, the process is essentially the same, except you don't have to capture anything because you want to sequence every region in the genome. And so this introduces an easier workbench or an easier wet bench workflow, but it also introduces less bias into what you're sequencing. So the advantages of whole genome sequencing over exome sequencing for structural variant detection are fewfold. Whole genome sequencing does not require probe enrichment like we talked about, and so you get less bias coverage. So if this is your exome on top with these black boxes representing exons, you'll get coverage only over your exons and you will not get coverage across the rest of the genome. With whole genome sequencing, yes, you will get some variability of coverage across your entire genome, but you will have some coverage across the genome, which can be normalized across these segmented windows. Another problem with exome sequencing is that GC-rich regions are very difficult to capture with exome sequencing, and GC-rich regions are pretty important because they are highly represented across the first exons of genes. So you'll often have a loss of coverage over those first exons, and so if you, for example, have a deletion that involves the first exon of a gene, you might actually miss that if you're trying to do your structural variant detection by whole exome sequencing. And then finally, pretty obvious, whole genome sequencing is contiguous. So you can identify the entire size and breakpoints of your structural variant, knowing exactly where the break is. And sometimes the breakpoint is critical because you could see alterations over the top of a couple exons, but it may actually be that the breakpoint falls in a critical regulatory region, and that could be what's clinically significant. So there are essentially 4 methods for structural variant detection from sequencing data. And we're using several of these. I'll start with the one that's probably the easiest to understand, which is read depth. And that's just simply looking at difference in coverage across the genome. So if you have your reference sequence, you have your little red and green sequencing reads down here that are aligned. And if you have a deletion, you would expect to have a loss of sequencing reads. Here, this would be consistent with a homozygous deletion because you have no sequencing reads. If it was a heterozygous deletion, you would have 50% of the sequencing reads. If it was a duplication, you would expect to have extra reads or extra coverage over this particular region. This method will detect gains and losses, deletions and duplications, but it will not detect anything that's balanced. Then we have read pair over here on the left, which analyzes the mapping information of paired end reads. And it's looking for discordance between what the expected insert size is between these paired end reads, or whether there's a discrepancy between the orientation as well. So read pair methods are really good for both. They're good for copy number, but they're also good for balanced rearrangements. However, they will not give you the exact breakpoint. That takes us to split read analysis, which is great for specific breakpoint detection because it takes reads that are overlapping a breakpoint. So if you have a deletion here and you have this read that overlaps your breakpoint, you have one of your paired end reads that maps to a region of the genome, but then in your other paired end read, only half of that read maps to the correct location in the genome, and the other half of the read is split and actually maps to somewhere else in the genome. So this will give you exact breakpoints. And then there's assembly, which we aren't using. Assembly methods are basically reconstructing the genome piece by piece de novo. and putting it back together. So that's great for identification of novel insertions, for example, but it's really something that works better on small genomes, and it is not great for repetitive elements, which are common within the human genome. So we are using a combination of the first three methods. And each of these methods has kind of their advantages and disadvantages. If you're looking for large CNVs, so things that would be detectable by microarray, your depth of coverage method is going to be the best for detecting those CNVs. However, it's really poor at breakpoint accuracy. So this is going to kind of be more similar to a microarray type of detection. Your split reads are great at breakpoint accuracy, but they're actually pretty poor at large CNV detection. And then in the middle are your discordant read pair and assembly methods. So you can see there's an advantage to having multiple of these methods used together because they really offer different benefits. The purpose of our project is to develop a clinical grade whole genome sequencing assay that includes detection of structural variants. So today I'm only talking about the structural variant portion, but we also are validating the whole genome sequencing for detection of single nucleotide variants as well as small indels, and then layering on top this detection of structural variants. And this is really for patients who have who would have an exome typically ordered, but in this case now they can have the added bonus of getting structural variants detected within a single assay. It's also beneficial for patients who have already had an exome testing that is negative, and for patients just with complex disease where, you know, if a patient has a very clear clinical diagnosis that's associated with a single gene disorder, you probably don't need to order this test as your go-to first line test. You know, it still makes sense to order your small panels for things that have a very clear clinical diagnosis. But a lot of patients are coming in with really complex disease and not having a clinical diagnosis, and that's where this test may make sense as the first test for some of those patients. So the validation overview is that we had 8 genomes that we sequenced. The samples that we chose had a clinically significant structural variant. I know a lot of labs validate their genomes with reference samples that don't necessarily have anything clinically significant, but we wanted to make sure that we could detect structural variants that we knew were clinically significant, and they had all been validated by an orthogonal clinical assay, so they either had microarray testing previously or they had a targeted panel that had detected a structural variant. We are sequencing on the NovaSeq at the UMGC. We're using 50x coverage, and we then are taking our call set and validating a subset of the calls, about 100 of them with qPCR, which is a clinically validated test in our lab. So this is just kind of the range of clinically significant structural variants that we were looking for. We're also validating some of the not necessarily clinically significant structural variants, but these are the ones that we wanted to make sure that we caught. And so we tried to pick a range of sizes here, down from something that involves a single exon up to things that would be visible by karyotype. We are using an algorithm called SpeedSeq for our structural variant calling. This is a combinatorial method combining split read, paired end, and depth of coverage methods for structural variant calling. The split read and paired end methods are combined into an algorithm called LUMPY. And then there is a kind of quality measure which allows you to fine tune your call set based on how much you require overlap between the split end and the paired end read calls, called SVtyper. And then it also includes our depth of coverage method called CNVnator, which uses that segmentation window size that I mentioned earlier. One of the advantages clinically is that it runs pretty quickly. This is something where we can't really afford to be kind of churning through our genomes in long periods of time, because they're often kind of fairly rapid clinical questions that they need to answer with this assay. And then we started by testing it on a reference sample that didn't have any known clinically significant structural variants versus an array result on that particular sample. And all the array structural variants that were not detected by SpeedSeq we're in regions that are already difficult to sequence or align because they have mapping issues or they're repetitive, and those are issues that we wouldn't expect to be able to interrogate with sequencing anyway and would be excluded. So we felt good about moving forward with SpeedSeq. So this is our filtering scheme. We're starting with our raw data, our BAM file. We're excluding those regions that are difficult to map, like we talked about. And there's kind of, in the field, you can find exclude regions or sets of areas of the genome that are repetitive, difficult to map, et cetera. We're never going to be able to confidently call copy number gains and losses there. That in our first sample, we looked at it in our reference sample, left us with 3,500 deletions and over 1,600 duplications, which is not a number that's feasible to interrogate clinically. So then we applied some quality filters and allelic balance filters, making sure that if it was called a deletion, that the allelic ratio makes sense for a deletion, for example. That left us with about 1,200 deletions and over 200 duplications. And at that point, we limited the calls to the exome plus or minus a 200 base pair pad. Because like I was mentioning in the beginning, most of the variants that are known to be clinically significant are going to overlap it with that exome. And particularly for structural variants where we have even less information out there in the literature, we're not going to really be able to interrogate deletions and duplications that are hiding out there in introns. So we're requiring just some overlap with an exon. It doesn't have to be contained within the exome, it just has to overlap. That left us with about 500 deletions and duplications combined, at which point we had people manually go through all those deletions and duplications and annotate what was going on with them. So our annotations, of course, whether it was a del, a dupe, what the size of the variant was, whether it was a double positive or single positive, meaning was it called by both LUMPY and CNVnator, or was it called by LUMPY only or CNVnator only, which algorithm called it, whether it overlapped with a curated list of about 4,100 genes that we know are clinically significant and we already interrogate on our targeted sequencing panels clinically, or whether it overlaps with a gene that is not known to be clinically significant, whether there are any mappability issues for that region and how much of the region of the call has a mappability issue, whether it is a false positive or maybe real. This is the most time-consuming part, which is a human being looking at the call and saying, does this region look real or not? Does this call look real or not? Based on whether there are mappability issues, how much of the region is overlapped with mappability issues, and whether the evidence, the LUMPY or CNVnator evidence, looks like it supports that call. So if you have a split read call set over that region and they all have slightly different breakpoints, then you're starting to be less confident. But if you see a LUMPY call and all the split reads are completely overlapped with the same breakpoints, that's better support for the call. Then we made a recurrent call list after analyzing all of our genomes, which are events that are either common in the population and happen to occur in a lot of our samples, or they are things that are technical artifacts, potentially repetitive regions. We looked at the population frequency based on NOMAD structural variant information, and we also can look at DGB for larger events. And then we just have an annotation of whether the region has been stitched. So if we have two calls that are very close to one another, and there's just an intermediate region that's not called, if they're very close to one another, they will be stitched together and called out as one call. And sometimes the reason for that is the region that's maybe actually overlapping with an exclude region or a repetitive region in the middle, and then that middle part will fall out of the call. So the goal of this annotation process was to essentially find categories of calls or types of calls that in the future we could just not deal with. We either could annotate them in an automated fashion so a person doesn't have to look at them, or we would not have to do qPCR confirmation, because feasibly there's kind of an upper limit on the number of qPCRs that you're going to be able to do with a clinical assay to make it both economical and time sensitive. So this is how we are visualizing our calls. This work has been done by Nathan Pankratz's group, developing the visualization tool. Here you can see this kind of mountain peak data is the coverage data. So you can see here where the coverage falls down. That's consistent with where our deletion is. You can see why the depth of coverage method is not perfect for calling the breakpoints. Then we also have our discordant reads here, which support this call of a deletion. And then you can see it was not called by the split reads down here at the bottom. Maybe it caught one of the breakpoints and not the other one. We have our gene list that it overlaps here. Actually, I have a zoomed in view. Our gene list that it overlaps, what the frequency of overlap is in Nomad. You can see there's a little bit here, but that's not overlapping with our call. What we would be really looking for here is like an entire deletion here that occurs at maybe 5% of individuals in Nomad. That would be something that's not clinically significant, common in the population we could toss out. And then the bottom is our mappability track for whether this region has a lot of mapping issues, and it doesn't, just little pockets here and there. So these are kind of the annotations that we are making, either making manual annotations or whether they're automated, what type of call it was. And then we can get a lot of information about whether it's in DGV, what the frequency is, what the Nomad overlap is, what the percentage of overlap of our call is with the mappability track, the size of the variant, etc. So this is a really kind of, nicely put together tool that allows us to get a lot of information quickly because we're going through a lot of structural variants and need to be able to weed out things as easily as possible. So, so of those calls that I showed you earlier, some that were detected by arrays, some that were originally detected by targeted panel structural variant detection. About CNVnator caught 100% of those by whole genome sequencing. And LUMPY caught about 70% of them. So all of our structural variants that we were looking for that were clinically significant were caught by at least one method using SpeedSeq. So that was great. And then we took those a subset of those calls and ran qPCR, not those same calls, but a different set of calls. So we took a different subset of the SpeedSeq calls and did qPCR, or we had the corresponding array. And there was only about 40% concordance for deletions and duplications between the SpeedSeq call and our qPCR or array result. Meaning that over half, so 58% of the calls that we get are actually false positives. And we don't really want to have to comb through all those false positives manually. So, so we looked at the concordance between our qPCR result and what we had annotated the call as when a person went through and looked at it using those criteria of mappability issues and lumpy support and things like that. And we found that nearly 60% of the deletions were correctly annotated as false positive with qPCR, and nearly 80% of the duplications, sorry. So this is real and false positive, sorry. So we were about 60% accurate with deletions and 80% accurate with duplications, calling something real versus false positive. So now I'll break it down specifically into false positive. So about 89% of the events the calls that we called false positive were actually correctly annotated. So we were pretty good at calling false positives. And 3 of them did show a true deletion by qPCR. However, all of those were common CNVs that are either difficult to sequence regions or repetitive regions. And so this led us to the conclusion that the calls that we're annotating as false positive, the criteria that we're using as false positive, are good, and we can trust that if we call something a false positive, it either is a false positive or it's a common CNV, which we don't really want to be looking at in the future anyway. Double positives that were called by both CNVnator and LUMPY, about half were normal by PCR and about half were real. However, all of the ones that were not confirmed by PCR had mappability issues, were already annotated as a false positive, or were common CNVs. So all of those that would have all of the double positive calls without mappability issues would be something that we would know was real, because they all confirmed unless they have these mapping issues or were annotated as false positive. So if we exclude false positives and common CNVs, we can trust that our double positive calls are true. Of lumpy only calls, most of them did not confirm by qPCR, and the ones that did confirm were all smaller than 2 kb. That's not surprising, because as we talked about, the split read methods are much better on the smaller scale and not so good on the bigger scale. And most of the ones that were normal were larger than 2 kb, i.e. if you have a large call greater than 2 kb without any depth of coverage support, it's probably just junk. So lumpy calls lumpy only calls greater than 2 kb without CNVnator support are not real and don't require further confirmation. CNVnator calls were hit or miss. Some did confirm by PCR, some did not. There was no trend on the size, whether it had mappability issues. So unfortunately, every call that's made by CNVnator by itself will require further analysis by, a genetic counselor, a pathologist, or if it's clinically significant, with follow-up by qPCR. And then calls, if we limited to overlap with the clinical gene, that were discordant with our qPCR result, most of the clinical gene calls, which would be the ones that we would think about reporting, most of them had mappability issues, were in that lumpy only greater than 2 kb category, which we'd be eliminating, were recurrent across our samples, which are either artifact or repetitive region, were correctly annotated as false positive. So what this leaves us with is out of our entire validation set, there would only be a couple events that there was no clear reason for the discordance with the qPCR result. So if we exclude recurrent regions, regions with mappability issues, greater than 2 kb lumpy only calls, false positives, then we would only have a couple calls that require qPCR confirmation, which is more along the lines of what would be feasible for a clinical assay. So our clinical workflow is to review all the calls that are double positive, that overlap a clinical gene, would be, reportable. Lumpy only calls that are small, that overlap a clinical gene. CNVnator only calls that overlap with clinical gene, we have no other way to limit those CNVnator calls. Double positive calls that do not overlap a clinical gene, this was our way of kind of interrogating some novel genes. We're not going to be able to look at every potential novel gene call. However, we figured that the double positive calls really tend to be true. And so there would be a lot of bang for our buck there to look at those and go into the literature and say, could this maybe be a new clinical gene? Is there any recent paper that has been reported for those? Then perform clinical variant interpretation, which I won't get into here, but is really the time-consuming part of the bulk of this work. Is having our pathologists and our genetic counselors comb through databases and the literature, trying to find any evidence that these variants are significant or not. And then for any variants that would be reportable clinically, to perform qPCR or breakpoint spanning PCR to confirm them, and then go to the clinical report. So our future directions are building of our clinical database to house these structural variants, which will help us with future clinical interpretation as well as for research, expansion of the visualization tool to make it more interactive, and then in the long run to develop long read sequencing, which will really be necessary if we want to do balanced events. So the methods that we're using now with short reads are not going to be sufficient for translocations and inversions, and that we will need to develop long read methods for. With that, I'll acknowledge the group. I did not do this effort at all by myself. Bharat is our co-lead on this project, as well as the rest of our clinical team here. Nathan's group, who has worked a lot on the visualization, and MSI, the Supercomputing Institute, who have done all of the bioinformatic work for us. So with that, I can take any questions.
Thank you so much. Yeah, and a little update, COVID update. We've got that dynamic interactive tool coming along nicely, and should have that for you soon. Big thanks to Caitlin for working on that, and Ben Cole, who did a lot of the development of that framework that you presented. Questions from the audience?
Hey, this is Joe. I have a question. How much computer science background do you have, and do you have any ideas for upcoming trainees and younger faculty members and how to accomplish some of that? Because this is really impressive. Great job.
Yeah, so my computer science background is 0. So I rely a lot on the folks from MSI and from Nathan's group, and they would be able to speak better to what kind of our opportunities in the future with trainees would be. I'm guessing there are great opportunities for trainees to work in that area. But my personal experience is nil. So I rely on a lot on the support of these people.
Yeah, I would just like to add, Dr. Lennox, that what we have tried to do with our Supercomputing Institute hires, the bioinformatic hires, is to have people who have some training in biology, in addition to a lot of training in computer science. And that's been a very fruitful way of integrating people who can work across disciplines. So.
Yes, you definitely need the expertise that you and Kelsey provide, and then the workhorses for coding, and you need somebody to interpret the two, do that crosstalk. We have a question from a member of the audience asking about MANTA and SVABA as another way to look at these. I've certainly looked at a lot in our research, and SVABA has had the best sort of mix of sensitivity and specificity. It's a newer algorithm. Kelsey, Bharat, any talk about other methods that you are considering?
So we haven't tried either of those. And it is possible going down the road that we would try other things, but like we heard in our talk before, once you kind of start to develop a clinical pipeline, you start to kind of lock it down. We could certainly go back and look at other things, but yeah, we haven't gone there yet.
Well, a big thanks for all 4 of our speakers for staying on time. And I want to take the opportunity to thank all our speakers. This was really a terrific symposium, and very informative to see what people at other places are doing and advancing what we do in molecular pathology. So thank you again to everyone who attended the symposium, and take care.